proteins, they are just these incredible, tireless workhorses of life, aren't they? Doing everything from, building our tissues to orchestrating these really complex biochemical reactions.
Absolutely essential.
But have you ever just stopped to think about the engineering? I mean, how did these astonishingly complex functional 3D structures just emerge from what starts as a simple chain of amino acids? It seems almost magical.
It does, but it's really about molecular geometry. It's physics and chemistry at its core.
Okay, so today in this deep dive, our mission is to sort of unpack the rules, the ingenious rules governing how proteins get their shape. We'll start right at the beginning with the first connection, the peptide bond. And then we'll see how this brilliant principle, visualized in something called the Ramachandran plot, helps us predict, well, their most stable and functional forms. It's quite a journey.
It really is. From basic chemistry to the blueprint of life.
Okay, let's get into it. So if we're building these molecular machines, we have to start with the very first link, right? How do the individual amino acids actually connect?
Right. It all starts with the peptide bond.
Yeah.
Fundamentally, it's a covalent bond. You take the carboxyl group of one amino acid, that's the carbon double bonded to an oxygen, connected to the central alpha carbon.
Okay.
And you link it to the amino group of the next amino acid in the chain.
And this happens inside our cells.
Exactly. It happens inside the ribosomes, the protein factories, really. This reaction is precisely controlled there.
And it's a specific type of reaction, isn't it? Something we see elsewhere in biology.
Absolutely. It's what we call a dehydration reaction, or sometimes condensation. Basically, a molecule of water, H2O, is released when the bond forms.
Right, like squeezing out water to link things together.
A good way to think about it. But beyond just forming the bond, there's something really fascinating with the peptide bond itself. It has this property called resonance.
Resonance. Okay, chemistry term. What does that actually mean for the bond, for the protein structure?
It's incredibly important. Resonance here means the electrons aren't just stuck between, say, the carbon and the oxygen. They're sort of shared across the whole peptide bond group, the C, the N, the O.
So they're spread out a bit.
Exactly. And this sharing gives the bond some characteristics of a double bond, even though we usually draw it as a single bond between the carbon and nitrogen.
Okay, partial double bond character. Why does that matter?
Because unlike a normal single bond, which can spin freely like an axle, a bond with double bond character is rigid. It's locked. It cannot rotate.
Whoa, okay. So that link between amino acids isn't a flexible hinge. It's more like a flat, stiff plate.
Precisely. The peptide bond unit is planar. All the atoms involved lie in the same flat plane. And to minimize atoms crashing into each other, what we call steric hindrance, the alpha carbons on either side of this rigid bond are almost always positioned trans to each other.
Meaning on opposite sides.
On opposite sides, yeah, like diagonally across the planar unit. This is much more stable than being on the same side, the cis configuration, where they'd clash. So the key takeaway for you here is this unexpected rigidity right in the backbone.
That makes sense for building a stable structure, a strong backbone. But proteins move, they flex, they bind things. They aren't just rigid rods.
No, definitely not.
So if the peptide bonds themselves are locked, where does the chain get its flexibility? How can it fold up?
That's the perfect question. And this is where it gets really interesting structurally. While the peptide bonds are rigid plates, the single bonds that connect each alpha carbon to the rest of the backbone can rotate.
Ah, okay. So the alpha carbon, the central carbon of each amino acid, acts like a swivel joint.
Exactly. Those are the points of flexibility. Think of the peptide bonds as the rigid links in a chain, and the alpha carbons as the pins connecting them, allowing the whole thing to bend and twist.
Right, like a bicycle chain. The individual plates are stiff, but the connections let it curve.
That's a great analogy. Yeah. So these rotations around the alpha carbon give the polypeptide its crucial degrees of freedom. And we measure these rotations using two specific angles. Phi, that's the Greek letter A.
Right, okay.
Hit the SI. Phi is the angle of rotation around the bond between the nitrogen and the alpha carbon. Psi is the rotation around the bond between the alpha carbon and the carboxyl carbon.
Got it. And alpha carbon is phi, alpha carbon C is CI.
Got it. And these two angles, for every single amino acid, essentially define the conformation of the protein backbone. They dictate how it can bend and fold into its final 3D shape.
So we have these rigid plates, the peptide bonds, and then these flexible joints, the phi and psi angles at the alpha carbons. Now, does that mean with all those potential twists, a protein could just, fold into any random shape imaginable?
Ah, good question. Seems like it might, right?
Yeah.
Infinite possibilities.
Yeah, or are there rules, like molecular traffic laws saying you can't turn that way? This feels important.
There are Absolutely rules. Really fundamental ones. And it comes down to a very simple principle. Two atoms cannot be in the same place at the same time.
Okay, basic physics.
Basic physics applied to molecules. Remember, each amino acid also has a side chain, an R group, sticking off that alpha carbon. Some are small, like glycine's hydrogen, but others are really bulky.
Right, like tryptophan or something.
Exactly. So if those phi and psi angles could twist into any combination, inevitably some of those bulky side chains, or even atoms in the backbone itself, would collide. They'd try to occupy the same space.
That steric hindrance thing again.
That's steric hindrance, yes. And it's not just awkward. It creates a high-energy, unstable state. Nature avoids those.
So like a molecular traffic jam that causes the whole system to break down energetically.
Precisely. And this leads us straight to the Ramachandran principle, named after G.N. Ramachandran, an Indian biophysicist who did pioneering work on this in the 1960s.
Okay, what did he propose?
He proposed, based on calculations, that when amino acids are linked up, the phi and psi angles won't just adopt random values. They will naturally tend towards the angles that cause the least amount of steric hindrance.
The path of least resistance, basically.
Exactly, the path of lowest energy. So depending on the amino acids involved, certain combinations of phi and psi angles are much more likely, much more stable than others. Some are favorable, some are just forbidden.
That's really elegant. The shape is dictated by just avoiding collisions. But how did they figure out which angles were okay back then? This is before we had detailed structures for lots of proteins, wasn't it?
It was, largely.
Yeah.
Ramachandran and his colleagues used theoretical models. They took the known lengths of chemical bonds, the standard angles between bonds, and the sizes of atoms, their van der Waals radii.
Okay.
And they essentially built little molecular models on paper or computationally. Then they systematically rotated the phi and psi angles and calculated, okay, if phi is this and psi is that, do any atoms clash?
Wow, painstaking work.
Incredibly painstaking. And the result of all these calculations is the Ramachandran plot. It's a graphical way to see which angle combinations are allowed and which aren't.
A map of stable shapes? What does it look like? How do you read it?
Okay, imagine a square graph. On the x-axis, you plot the phi angle, typically from negos 180 to plus 180 degrees. On the y-axis, you plot the psi angle, also negos 180 to plus 180. So every possible combination of phi and psi for an amino acid residue corresponds to a point on this graph.
And what did the calculations show on this graph? Was it just random dots?
Not at all. That's the beauty of it. Large areas of graph are completely empty. These are the forbidden regions where steric clashes would occur. Atoms would bump into each other.
So the white space means no go for the protein backbone.
Exactly. But then there are specific, well-defined regions on the plot where angle combinations are allowed, where there's minimal steric hindrance, and the conformation is energetically stable. And amazingly, Ramachandran's theoretical plot showed two major regions of stability.
Two main allowed zones. And did these zones correspond to anything known about actual protein structures?
They corresponded perfectly to the most common types of secondary structure that were just being discovered around that time, the fundamental building blocks.
You mean like the alpha helix and beta sheets?
Precisely those. One major allowed region, typically shown in the lower left quadrant of the plot, corresponds to the angles found in right-handed alpha helices. That tight coil structure fits nicely within these allowed rotational angles.
Okay, so the alpha helix lives in one zone on the map.
Right. And the other major allowed region, usually in the upper left quadrant, corresponds to the angles found in beta strands, those more extended zigzag structures that form beta sheets.
Incredible. So the theory predicted the reality of these common structures just based on atoms not bumping into each other.
Exactly. It showed how these fundamental secondary structures aren't arbitrary. They are the natural consequence of the allowed geometry around the alpha carbon, dictated by steric constraints. There are a few other smaller allowed regions too, corresponding to things like left-handed helices or turns, but those two are the big ones.
So beyond just predicting these structures, how is this plot actually used today? Is it still relevant in research?
Oh, absolutely indispensable. It's a fundamental tool in structural biology. For example, when scientists solve a new protein structure using experiments like X-ray crystallography or cryo-EM, one of the first things they do is check its Ramachandran plot. They plot the phi and psi angles for every amino acid residue in their structure. If they see a lot of residues falling into those forbidden white regions, yeah, it's a big red flag. It suggests there might be errors in their experimental model, that the structure isn't physically realistic. It's a crucial quality control check.
Makes sense. Like proofreading the structure against the rules of geometry.
Exactly. And it's also vital in things like drug design.
Yeah.
If you're designing a molecule, maybe a peptide mimic, that needs to bind to a protein, you need to make sure the backbone shape you're designing is actually achievable, that it fits within the allowed regions of the Ramachandran plot. You can't design something that forces the protein or the drug into a sterically impossible shape. It helps guide rational design.
And maybe understanding diseases where proteins misfold?
Definitely. By analyzing the Fibsy angles in normal versus misfolded proteins, researchers can sometimes pinpoint the specific conformational changes that might lead to aggregation or loss of function, like in Alzheimer's or Parkinson's disease. It gives clues about the problematic shapes.
So it really connects the basic physics to structure, function, and even disease.
It absolutely does. It shows how these quite simple physical rules end up defining the complex world of biological structures and provides a really powerful way to analyze and even engineer them.
What an amazing journey, then. We've gone from the surprising rigidity of that peptide bond
Right.
through the specific freedoms allowed by the phi and psi angles, all the way to the Ramachandran plot, which basically maps out the stable shapes based on just not letting atoms collide. It beautifully predicts the alpha helix and beta strand, the core components of almost all proteins. It really is a blend of chemistry and pure geometry.
It is. And it highlights this, profound elegance, how incredibly complex biological machines emerge from just a few fundamental physical laws. Life still has to obey the rules of physics.
Absolutely. Which leaves a really interesting thought for you, our listener. Think about all the complex machinery inside you right now. Enzymes, antibodies, receptors, motors. How much of their specific functional shape is ultimately dictated not by some complex biological code alone, but by these very simple, elegant rules of molecular geometry, avoiding clashes, finding the lowest energy state, all visualized in that one quite beautiful chart. Something to think about next time you consider the amazing world of proteins.